Greetings, ladies and gentlemen. Professor Recore here to bring you the first in a multi-part series of tutorials describing muscle contraction. In this particular video, we're going to be talking about skeletal muscle contraction, and we're going to be specifically looking at the sliding filament theory of skeletal muscle contraction to describe what happens at the ultrastructural level. So let me first define the scope of this particular presentation. So we need to remember that muscle contraction involves a number of specific events that have to take place in sequence, and that detailed discussion of these events merit their own presentation. We will be talking specifically about skeletal muscle contraction via the sliding filament theory at a specific place in the sequence. And let's take a look at that place right now. So you can see on the left where it has phase 1 and phase 2. You can think of muscle contraction steps as starting at the very top in the peach/orange colored box and then moving downward as the steps go along. Well, the steps that we're going to be specifically talking about in this video are the last two steps in phase 2. Specifically, the portion where calcium binds to troponin and the myosin binding sites on actin are exposed, and the myosin heads bind to actin, allowing contraction to begin. This occurs at the ultrastructural level of the myofibril, which is known as the sarcomere, which is one of many terms that we'll get into here in just a second. But I just wanted to define the scope of the presentation so that you all know exactly what we're going to be talking about and how it fits into the overall concept of muscle contraction. Condensing the series of multiple steps in muscle contraction that we saw on the previous slide down into two overall phases. Phase 1 would represent activation, and activation occurs at the neuromuscular junction. And this is activation that occurs via the nervous system, stimulating an action potential to occur at the sarcolemma, or the muscle cell plasma membrane. Phase 2 is excitation-contraction coupling, and it's within this phase that the steps exist that we are going to be discussing today. So first, let's discuss some terminology. Myofibrils can be thought of as densely packed rod-like elements that comprise approximately 80% of the muscle cell volume. And these myofibrils, they contain sarcomeres within them. And the sarcomere is the contractile unit within a skeletal muscle myofibril. These sarcomeres contain myofilaments in them. And these myofilaments exhibit striations. The striations you can even see here on the picture in front of you, these alternating light I bands with the dark A bands. Okay, let's take a look at a sarcomere up close. The Z-disc is a coin-shaped sheet of proteins on the midline of the light I band, and it anchors the thin filaments and connects myofibrils to one another. Next is the M line. The M line is a line of protein that's called myomesin, and it bisects the H zone, which we'll see the H zone in the next slide. Next, we have the sarcomere, and the sarcomere can be defined as the region that runs between two successive Z-discs. So from Z-disc to Z-disc, everything in between that is represented by the sarcomere. And remember, the sarcomere is the contractile unit of skeletal muscle. Next, we have the thick filaments, or the myosin filaments. The thick filaments run the entire length of an A band. The thin filaments run the length of the I band and partway into the A band. You can see that the thin filaments, the actin filaments, are represented by the color blue, and the thick myosin filaments are the ones in that orangish red color. One of the terms that we haven't defined are the elastic filaments, which are made up of the protein titin, and those are represented in yellow. This slide gives us a much better view of the H zone, and the H zone is a lighter region in the midsection of the dark A band where filaments do not overlap. To put into context, this slide represents a fully relaxed sarcomere of a muscle fiber. What you see in black and white, this is known as an electron micrograph, and this is one of the most detailed views that we can get in science using this electron micrograph technology. And we will be able to see how the view changes when the sarcomere is contracted first fully relaxed versus fully relaxed, as we see right here. So bear with me just a few more slides with definitions, and then we'll get into the sliding filament theory. So recall that actin composes the thin filaments and myosin composes the thick filaments. And the orderly arrangement of this actin and myosin is what gives skeletal muscle its striated banding pattern. So the thick filament is composed of the protein myosin, and myosin heads actually contain these two smaller polypeptide chains that act as cross bridges during contraction. Moreover, the myosin heads serve as binding sites not only for the actin of the thin filaments, but also for the ATP. And the ATPase enzymes, which is simply a way of describing the enzyme that phosphorylates or breaks ATP down, allows ATP to be used, are functional here. In the thin filament, one thing to make sure that we know is that tropomyosin and troponin are regulatory proteins that are bound to actin. And we're going to be talking about how we're going to have to move them off of actin's binding sites in order to allow for the sliding filament theory to occur, in order to allow for contraction to occur. Let's take a look up close at the thin filament and the troponin and tropomyosin that we were talking about in the previous slide. So I'd like to draw your attention to this bottom right graphic you see right here. And I want you to notice how the tropomyosin is sort of referring to this long brown colored helix shaped almost line of proteins that is blocking almost uniformly, blocking the active sites for the myosin attachment on the actin subunits. This is tropomyosin. And in yellow here, we have the troponin. What we're going to talk about in just a couple of slides is how the influx of calcium associated with the steps in muscle contraction are going to allow for calcium to be present to bind to troponin. And when calcium binds to troponin, what that allows is for the tropomyosin to be shifted off of those active sites on the actin subunits for myosin attachment. And this will allow the cross bridges to form and myosin to bind to actin in our sliding filament theory. Our sliding filament model of contraction tells us that during contraction, the thin actin filaments slide past the thick myosin filaments, and this causes actin and myosin to even more overlap than they were before. And cross bridges form when the myosin heads bind to actin. When this occurs, sliding begins, and cross bridges form and break several times. And overall, this is going to cause shortening of the muscle fiber. The Z discs will be pulled toward the M line. The I bands will shorten. The H zone disappears, which is that part right in the middle, and the A bands move closer, even though their length stays the same. So in front of us, we have the representation of our fully relaxed sarcomere of the muscle fiber. Notice that our H zone is nice and neat in place and in the middle, and our Z discs are along the sides and with the I bands below it and the A band in the middle. Next, we're going to take a look and see what it looks like when the sarcomere is contracted. Notice how that H zone has disappeared and the Z discs have also gotten shorter. In this fully contracted sarcomere, our myosin and actin filaments overlap even more than they did so during relaxed state. I just want to remind everybody of specifically where in the contraction cycle we are again. And we are at the point where the sarcoplasmic reticulum has released large stores of calcium. And now we have that free calcium that's available to bind to troponin. Doing so causes the tropomyosin to shift off of those myosin binding sites on actin, which is another way of saying that those binding sites have been exposed. Exposure of those binding sites allows the myosin heads to bind to actin, allowing contraction to begin. So let's take a look at that. You can see here again, we have our actin in blue. Our tropomyosin is that brown molecule that's blocking the active sites, and the troponins in yellow. When calcium binds the troponin, this removes the blocking action of tropomyosin and exposes those myosin binding sites. Since the myosin is allowed to bind to actin, this forms cross bridges and contraction as well. This is known as cross-bridge cycling, and that's what begins. And at this point, excitation-contraction coupling is over. This brings us to the last portion of this part of muscle contraction, which is to talk about the steps involved in the cross-bridge cycle. Step 1, the cross-bridge formation. This is where that energized myosin head attaches to the actin myofilament, and in doing so, a cross bridge is formed. Step 2 is known as the power stroke. This is where adenosine diphosphate and inorganic phosphate are released, and the myosin head pivots and bends, changing to its bent low energy state. As a result, it pulls the actin filament towards the M line. In the third step is cross-bridge detachment. After ATP attaches to myosin, the link between myosin and actin weakens, and the myosin head then detaches, which is another way of saying that the cross-bridge breaks. Cocking of the myosin head is the fourth step, and the cocking of the myosin head occurs as the result of hydrolysis of ATP into ADP and inorganic phosphate. This causes the myosin head to return to its high energy conformation, or that cocked position. Finally, no presentation by Professor Recore would be complete without some kind of clinical correlation. And in this case, we're going to be talking about why rigor mortis occurs. Well, rigor mortis, which is the stiffening of a cadaver after death of the human, this occurs because of the lack of ATP in the person who has deceased. The lack of ATP causes an inability for the cross bridges to detach, and therefore myosin stays bound to actin. And long story short, the muscle is unable to relax. And that's what causes that muscle contraction that is commonly referred to as rigor mortis in a cadaver. So that completes this tutorial on skeletal muscle contraction via the sliding filament theory. I'm Professor John Recore, and I appreciate you all sticking it out with me and hope you have a wonderful day.